your chemically defined, which you have to have the exact, it's very expensive, but certain ones like it. Complex is cheaper, and it's just a whole bunch of different complex items that they can put in there. Selective, this is the one where it suppresses one and makes another grow. Differential makes it easier to distinguish colonies. This is where you got your alpha, beta, and gamma of your strep. Alpha is almost, beta is best, and gamma is
no good.
no good.
Hemolysis.
No, but I believe we got selected because that was the fungi and bacteria. That one.
What is lichen?
Ooh.
It's not a werewolf.
No. No, that'd be kind of cool.
Close.
Close?
Algae and what?
Close.
Yeah, it's close.
Oh.
Sorry.
I'm like,
Close.
geez. Doesn't it like grow on rocks? I don't know. Tree,
Well.
it's green. I don't know.
Oh.
Is that right?
It does grow on trees. What?
Bacteria?
Say it one more time, you cut off.
Cyno or cyanobacteria? I don't know.
Good guess. It's algae plus something.
Oh! Algae and fungi? Algae and.
Is it?
I think it might be.
Red algae. Isn't it like red algae?
Yeah, it's
And
fungus.
It's
Fungus.
green algae.
Green
It's
algae.
green algae and fungus.
Oh, okay.
Sorry, I just remembered that, so I thought I'd better get it out before I forget it. Okay, what are phases of growth, and when would it be best to use penicillin?
The lag, log, stationary, and death.
Very good. When would we use penicillin? Why?
Log phase.
Good job, it's the growth phase, right? Good job. What kind of bacterial oxygen requirement is set for tetanus? Remember the foot? Here's your foot. Now it goes inside the foot.
Yeah, so it's not an aerobe.
Right.
It's anaerobe.
Good job, an obligate anaerobe.
Yeah.
These are the ones, I would know these because I'm sure he'd have them in a question, if not a question itself, but definitely in one. You've got obligate anaerobe, so it's against air, so it's like air. Facultative anaerobe, this is the one where I think of faculty. Faculty is everywhere. They're in the classroom, they're outside the classroom, they're breathing, they're not breathing. They're everywhere, faculty. Obligate aerobe, so they have to have air. Atolerant anaerobes, so they don't need air, but it doesn't hurt them. So with obligate anaerobes, it would kill them if they were in air, but these, they're fine with air. They just don't like it. And then micro aerophilize, I cut this up. You got micro, which is little, aero, which is air, philize, likes. So it likes little air. So there's only little air that would be present. So what kind of a bacteria would be a facultative bacteria? Where would you find something like this? Or an obligate anaerobe? Where would you find those two? We just talked about it.
Tetanus?
Okay?
No.
That would be one.
Yeah.
That would be one. You're not wrong. These would be found in the GI tract mostly.
Oh.
Remember? What is the best way to check for bacteria in sewage? And what are some ways to count?
Enterotube?
Very good.
Enterobe.
Enterotube is a very good one. Standard plate count or filtration is another one.
These are how you count.
These are how you count them, but filtration could be one. I didn't put that arrow too, but it's definitely right. So turbidity is you're just looking at to see the cloudiness. Direct electron counting is where something counts it, and you don't know if it's dead or alive. Same with direct microscope. This is where you take a swab of sample and you put it on, and it has a little chart, and you count how many are in the chart. You don't know if they're alive or not. Standard plate count and filtration. Alright, what does it mean when it says mutagenous? Mutagenous. Mutagenous.
Like mutation of the DNA.
Is this good or bad?
Bad for us, good for
Very
Nice.
It could be both, couldn't it?
May be caused by recognizing an agent or chemical process called mutagenesis. It gives us these things, but whether they are good or bad will depend on what they are used for. So you're both right. So you got the nitrogenous acid, you've got benzo, whatever. You've got these. Radiation. What is DNA molecule made up of? Good job. What is the shape of it called?
Double helix.
Double helix. What is the genetic code pairing?
AT, adenosine and tyrosine, and then
I always remember they met.
AT. Yeah. I just remember
what was the
ATCG.
molecule name?
Oh man, this sucks. I always remember they met at G meets C. They met at GMC. These are the two where they match, okay? They are always, A is always gonna be with T, G is always gonna be with C, whether it goes CG or TA, it doesn't matter. Oh, that's a bummer. Oy. What is the process where mRNA, tRNA, and rRNA are used, and what are they used for?
protein synthesis?
You're making proteins, very good. What is an mRNA or messenger RNA? What does that do?
Doesn't it take information out of the nucleus and take it to like the ribosomes?
Very good. Okay, what does the tRNA do? We'll skip that one. What does the rRNA do?
What does it stand for?
I think it's ribosomal.
Proteins.
So you're right with the blueprint. Takes it to the ribosome. This is called transcription. Because you're writing it down. It's like a transcriptionist. They write down stuff from the doctor. I don't think he really talked about RNA, so we're gonna just kind of skip that one. But we'll go to tRNA. Okay, this is the translation. You're decoding it. So you're translating it. You're talking about it. You translate the transcription. Make sense? Okay, this creates the polypeptide chain. What is genetic recombination? This is the exchange of genes between two DNA molecules to form a new combination of genes on a chromosome.
They can do that, can't they? Like, can't scientists do that to manipulate like what they want?
To a certain point, I think they
A
can.
little bit, anyway.
Yeah. Didn't they, is this the one where they hollowed out the virus and they put in there and had the virus like send it to the certain cell that they wanted to, but it didn't work out really well? I can't remember what that's, I think, don't quote me on that one. Can't remember that one. Anyway, what are some different types of genetic recombination? So you got transformation, when the genes are transferred from one bacteria to another, as naked DNA in the solution. You got conjugation, when genes are transferred from the donor bacteria to the recipient. This is the sex pili. the transduction, when genes are transferred from a donor recipient by the bacterial, yeah, so that's bacterial virus or bacteriophages. No, it's not. Transduction, okay. Transporsons, or transpors, whatever, are small fragments of DNA that can be moved from one region of the DNA molecule to another. This is the one where I think that they could manipulate a little bit more, where they could cut it off, move it where they wanted to. That's the one I think anyway. What do you think?
Sounds right.
Alright, and we're going on. You already know what a polyribosome is. We already talked about that. We're good. Sweet. Okay, let's read and go through some questions. A bacteria has an optimum growth temperature of 37 degrees Celsius. What is the likely host?
Us.
Humans. Very good. True or false? Thermophilic bacteria endospores can survive industrial canning. True or false?
Thermophilic, is that so?
Thermophilic bacteria endospores can survive industrial canning.
This is a trick question. True.
It's true. Good job. True or false? Microbial growth means the cell wall will grow bigger. Why? How does it grow?
Like that.
So there's more of them. It doesn't grow bigger, it just grows more. Good. Plasmolysis means that the bacteria dies. True or false?
Eventually, yes, right? Because
True.
the membrane is.
The shrink?
It shrinks, but if it's a strong bacteria.
Oh, it could put it into like a cyst or a spore, couldn't it? And it could
Survival
just go,
mode.
like.
Very nice. True or false? Halophilies love salt. Halophilies. Those things that mean. True. Okay. True or false? When you wash your hands, it kills all the bacteria. False. True or false? Organic compounds are only required by humans.
False.
False. Good job. Because they like to steal iron and things.
Oh. Stupid.
Anyway, 70% of all human bacterial infections involve what? 70% of all human bacterial infections involve what?
Biofilm?
Biofilm, very nice. Blank, blank cannot use oxygen for growth, but they tolerate it fairly well. Blank, blank, what?
Wait, repeat it once more.
Okay, blank, blank cannot use oxygen for growth, but they tolerate it fairly well.
Aeros. Oh yeah, there's
Yeah.
tolerant.
Aerotolerant. Aero?
Anaerobes,
No,
good job.
Anaerobes.
Aerotolerant anaerobes. Okay, we've already done that one, Okay, why would microbes be all over the agar as well as on top of the test tube? Why would microbes be all over the agar as well as on top?
They're aerobic?
Are they? What goes everywhere?
Facultative.
There you go, good job. Okay, why would microbes only grow on the top of a test tube?
Because they need it there.
They're aerobes.
Yep, obligate aerobes. Okay, let's see. A common and cheap agar. Anyway, the chemical composition is common and cheap, but it will vary. So the chemical composition varies. It's cheap and it's common. What is that called?
Hinton? Is it the Mueller Hinton one? Is it a type of that?
It's a type of agar.
Complex.
Complex, very good. Okay, blank blank media are exact chemical compositions and are very expensive.
Chemical.
Chemically defined. Okay, this media makes it easier to distinguish a colony of bacteria.
The differential.
Differential, very good. Let's see. Blank can be used for short-term storage of bacteria.
Fridge?
Fridge, good job. Blank blank set at 50, minus 50 degrees Celsius to minus 95 degrees Celsius is good for years of containing bacteria. Blank blank at minus 50 degrees Celsius to 95 degrees Celsius, negative, is good for containing bacteria for years.
Dry ice,
Deep freeze.
deep freeze.
So you can keep it.
Okay, blank is where you freeze dry the bacteria, which means that you take away all of the blank. Blank means, blank is where you freeze dry the bacteria, which means that you take away all of the blank. Starts with an L.
This is one of those terms that he might ask for.
Yeah.
It's like a special word.
Blank is where you freeze dry the bacteria, which means that you take away all of the blank. Starts with an L. Lipolysation. When you're taking away all of the water, it's like those army packets or their astronaut packets. Let's see. We already talked about that one. Talked about that. Blank phase is the phase that the bacteria stops growing because the nutrients are almost all gone.
Stationary?
Stationary, not blank yet. Blank phase is when the body is taking away some of the dead bacteria. Blank phase is when the body is taking away some of the dead bacteria. It's kind of a trick question. Death phase, good job. Okay, let's see. To find the original no, that's not gonna work. Let's try another one. Blank blank count is where you take the original inoculum and it's diluted, and then each dilution is cultured on an agar plate to get until you get to 30 to 300 cultures or colonies on the plate. So blank blank count is where you take the original bacteria that's inoculated in water, you dilute it by, I think it's 10 times each time, right?
I think so.
I think so.
Yeah.
10 times each time. Anyway, you dilute it, and then each dilution, you take a little swab and you culture it on an agar plate. And then you have to keep doing that until you get a culture where there's 30 to 300 colonies. Do you remember what that's called? What?
Something
Who would have thought? It's blank blank, so there's two words.
Standard plate?
Very good. Standard plate count. Very nice. Okay, let's see. I don't know if we talked about him. We'll do it anyway. Blank blank helped reduce infections associated with childbirth by washing of hands. Blank blank helped reduce what? Nope. He did the
Yeah, this one was
phenolic
in lab.
surgery.
He talked about it in lab.
Yeah, he did. He asked a question on it too, I think.
Is it Ignace?
Yeah, who named your child that?
I don't know, poor kid.
What's Ignaz?
Well, I think he was Austrian, so.
Ignaz Semmelweis. Oh, okay. Well, that
Semmelweis.
makes more sense. Ignaz Semmelweis. It's spelled like this. I don't know how to say it.
Semmelweis.
Could you imagine?
No,
That would just be
just
horrible.
exploding into
I
a bunch of
know.
different.
Different little companies.
Yeah, that would be weird.
Okay, anyway, what insect causes the African sleeping sickness? And what causes Chagas disease?
It's like the Tzet's fly or something.
Tsetse fly.
Tsetse.
Yeah.
So it's the tsetse fly and the kissing bug. I would not want to be kissed by that. Okay, what is an archaeozoa? What are two examples of pathogenic archaeozoas? Or archaeozoan?
ones that, like, live in extreme conditions or no?
No.
Oh, no.
So you're looking at like Trichomonas vaginalis, okay, so there's no cyst stage here. But you have Giardia lamblia, and that has to have a cyst stage, because if you drink the water, it's out in the water, it's gonna have to stay there for a while. The fecal sample of the what? What we're gonna, or what mammal makes this?
Beavers.
Beavers, there we go. So it's a eukaryote that lacks a mitochondria. Why is a mitochondria important?
It makes its own energy.
Good job.
Right?
Okay, how's Trichomonas vaginalis diagnosed? It's the wet mount test to see if it's motile. Plasmodium that causes malaria by the, wait, what was the other question? It's kind of weird. Oh,
Oh,
what are
3
three
examples.
examples? Sorry, totally missed that question. What are 3 examples of the, I'm not gonna try it.
AP complex.
AP complex.
Plasmodium.
Obviously. Okay, so you got plasmodium, what's another one?
Toxoplasma gondii. Wait, that's the cat, right? Okay, yes. It is.
Yeah, that's the one where they go crazy. Like you literally, like get schizophrenia from the cat feces.
That would be so sad. That's why people have, like, the crazy cat lady. She's
On
really
Simpsons!
crazy.
Yes.
It's true.
That's where it
She's
comes
all over
from.
cats everywhere.
I know, I love that.
I know, it's great. Okay. Cryptosporidium, where do you find that?
In contaminated drinking water?
Like, we had an outbreak up in Logan.
Oh.
Swimming pools. Very nice.
Was that at the aquatic center? Or something like that?
I don't know, I just know that my sister got it. She lives in Midley.
Ooh.
So,
I had somebody come up and visit, and they stayed at a hotel, and there was a big sign at the hotel saying, you know, be careful for cryptosporidium.
It was in the drinking water, not just like pool water. It was in the drinking water?
No, it was sorry,
No,
it was
no,
at the
in
swimming
the pool
pool
water.
at the
Oh, okay.
at the
Pool
hotel.
water.
Wow.
Sorry.
Yeah.
Yeah.
Should have said that.
Yeah.
Okay.
But
Wow.
yeah, so it was pretty much everywhere. It was just a big
That
outbreak.
Sucks.
Yeah.
She was so sick. Whatever. Okay. How do you classify protozoan?
By the worms that they are round or flat? No?
These are
Oh,
worms.
oops.
These are protozoans. Well, this is like your, what's a protozoan? Jock itch? You have ringworm?
Those are fungus.
Ringworm is not, it's a protozoan. I know that's a protozoan.
Okay. I believe you.
Jock itch might not be though. But I know ringworm
Tinea
is.
curris. Yeah, I think that's it.
Yeah.
Um,
I know
protozoa.
what it is. I went to the doctor. Unless the doctor's wrong, the doctor's
No.
never wrong. It's just like, it's never wrong. Right. Okay, anyway. We classify them based on motility and how they reproduce. If they have a cyst stage or not, and there are RNA sequencing. Mostly, since it's just kind of an overview, I would know its motility, sex, and cyst stage.
Like Trichomonas vaginalis.
Yeah.
That's one.
Oh, we just talked about that.
Mm-hmm.
Okay, well, protozoan causes amniotic dysentery, and how do they do this? How do they move?
I remember this is on, this is Doubtfire. Have you ever seen that?
A long time ago.
And the dad tells the little girl, your daughter has an amoebic dysentery. She's like, what's an amoebic dysentery? And I remember I learned it. I was like, ew, mucus. It's mucus. Like, oh, it does.
They move by extending blunt, like, oh, yeah, this is the blob. Yeah, this
Oh,
is how
yeah,
I
yeah.
remember, like, the
Uh-huh,
blob. being made
pseudopods.
The teacher, do you remember that? It was a really old book. I'm dating myself. But there's a blob that went to school and he ate the teacher and he would move like this. It was in a book, so that's why I always remember it.
That's funny.
Interleaved histolytica. What is a vector?
It's something that be like a fly.
Yeah.
That like moves.
Is an animal carrying a pathogen from one host to another. Very good. Okay. This is a passive transport of pathogen on insects' feet or other body parts.
Mechanical.
Oh, well, that wasn't right. I'll try that one again. Mechanical. There you go. Wow, that was weird. Something that a healthcare worker passes to the patient that infects and or kills the patient.
Not nosocomial, but it's the other one, right?
The one that starts with an I.
Isogenic or something.
Oh.
Yeah.
I don't know. It looks like that.
Iatrogenic.
Iatrogenic.
Why is it doing this? That's weird. Something that the healthcare, we already went through that. Stupid. What is the transient flora?
It's like there for a few days or weeks and it's gone.
Very good. What is the combination of green algae and fungus?
Lichen.
Where's it found? You got this one right. Where's it found?
Rocks and trees or something.
And why is it useful? It stabilizes the soil in dry and arid lands. I remember this. I remember one of those. I don't know if it was on a final or what, but.
Yeah, I remember that somewhere. Like, we might have had to write it in or something.